All right. What do you think, Mike? Should we start?
Yeah, I think we can make a start. Thank you all for joining us. Yeah, and I'll let you introduce the poster and Vincent.
Okay. Hello, everybody. Thank you for taking the opportunity or giving us the opportunity to present the data here, a cooperation poster from Reaction Biology Europe and from 4HF. My name is Jan Ehlert. I'm head of the cellular drug discovery or pharmacology team here in Freiburg. We have done the biologics part, that is the proliferation assay for 140 cell lines in our so-called ProLiFiler format, where we test 140 cell lines every, let's say, 6 weeks on a regular basis, and where we cooperate with our bioinformatics partner 4HF, also located in Freiburg, Germany, and who are analyzing the data that we generate as IC50 values and put them in their database and developed the algorithms to make the analysis that the Chief Scientific Officer of 4HF, Vincent, will now introduce to you. Okay, it's your stage, Vincent.
Thank you, Ian. So I am Vincent Varocco, the CSO of 4HF Biotech. Our company was founded in 2015 by Professor Fibic. And our company specializes and dedicated in the research with cancer data mining and in silico analysis. We have two main areas of activity. One is to support in silico analysis and to support the drug development with analysis of omics data. And the other aspect of our activity is the identification of novel targets to develop novel drugs for cancer treatment. So today, I will present to you the poster that we have developed in collaboration with Reaction Biology for the ACR 2021. So the poster is available. You can see it, right?
Yes.
Yeah.
Yes.
Thank you, Mike. So this poster is about and present the ProLiFiler and the cancer data miner that are tools for preclinical study to characterize novel anti-cancer agents. These platforms are dedicated to several things. One is to determine the potency of novel drugs. It's also to reveal the potential indication of the drugs, to define or to refine its mechanism of action, and to identify predictive biomarker. So for that, the analyses are done in 3 main steps. The first one is to test the anti-tumor potency of the compound in a large panel of cell lines covering most of the common cancer types. The next step is to compare the drug results with the respect of those of on-drugs dataset that we have in our database with defined or confirmed mechanism of action. It allows to investigate the mechanism or to reveal the mechanism of action, but also to compare compound activity. And finally, our analysis connect the drug data to genetic background and basal gene expression levels of the cell line to reveal predictive biomarker. So in this poster, we test and validate this approach. And for that, we have investigated 3 compounds. One is the BI-3406, which is an inhibitor of SOS1 KRAS. The second one is Nuclein 3A, an MDM2 inhibitor. And the last one is tacelicib, a PIK3CA inhibitor. So first, we will have a look to the results we have obtained with the ProLiFiler. I guess it's large enough like that. Yeah. So here where I show on the first scatter plot the results that we obtained for the nuclein 3A. This scatter plot, you have in X the drug potency, the IC50 value in micromolar, going from to the left, from the most sensitive to the most resistant on the right side, across the different cancer type on the white axis. And in bracket, you have the number of cell lines that were investigated per tumor type. What we observe for nuclein 3A is that this compound has a moderate potency and a strong selectivity with only few cell lines that was sensitive to the compound. If we take a cutoff of 1 micromolar for the pharmacological relevance, let's say, we can see that only few cell lines have an IC50 below this 1 micromolar. And finally, we can see also that this compound was active in many cancer types. This profile contrasts with the second one that I will present, that is for tazelicib. So for this compound, we have observed that this compound is much more potent with a median IC50 below 1 micromolar. The geometric mean is 0.5 micromolar. What we have seen is that the activity of tazelicib is for a wide range of cell line. And we have really sensitive differential activity that was seen in certain cancer types, such as leukemia, lymphoma, and also in some solid tumors like breast, non-small cell lung adenocarcinoma, or ovarian cell line. For BI 3406, the results were more tricky, and in fact, in our experimental condition, we obtained only a very weak potency of the compound with only an activity seen only in some cell line. And therefore, in the next part of the poster, I will not discuss and comment the analysis for this compound. So first, we will have a look to the MOA finder and the result that we can obtain with the MOA finder. So as said previously, it's an analysis that correlates the drug sensitivity result, the IC50, that we obtain with the ProLiFiler to those of hundreds of other drugs. In our database, we have more than 700 drugs for which we have annotation of the mechanism of action, and they are tested in more than hundreds of cell lines. The results of this analysis can be visualized with this scatters plot that have on the y-axis the raw coefficient of correlation on the level of significance in Y. So here, these are the results that we obtained for the nuclein 3A compound. And we also have annotation in the table below that show the top 8 compound that best correlating with the nuclein 3A results. And we can see that the top correlating drugs are all MDM2 antagonists. It includes in first place nuclein 3A that were generated in other datasets than this one, but it also includes the Sanofi compound, for example, the MI773 or the RG7112 that are also a nuclein an MDM2 inhibitor, and these drugs have really the same mechanism of action than the nuclein 3A. So it shows that the MOA finder was capable to retrieve the mechanism of action of nuclein 3A. Interestingly, in this analysis, we also found one of the compounds well correlating with nuclein 3A. It is the bortezomib. Bortezomib, it's a proteasome inhibitor. This compound is known to stabilize and activate p53. At the end, it gives the same inhibitory results than with the MDM2 inhibitors. So activation of the p53 and apoptosis of the cell, which explain the strong correlation between these two classes of compounds. For tazelicib, the analysis showed similarly to what we have observed for nuclein, is that the MOA finder allowed to retrieve the main mechanism of action of the tacelicib. Among the 8 top hits that best correlated with our tacelicib results, we retrieve only small molecules that target the PIK3CA signaling, and in the first place, the tacelicib coming from another dataset. Yeah. Right. So in total, for the MOA finder, we have shown that the MOA finder is capable to retrieve the drug mechanism of action, but it also allowed to validate that the data generated with the ProLiFiler well match with those previously reported in other study. So next step for our analysis is to search for predictive biomarker. So biomarker associate with response with sensitivity or resistance to the compound. For that, we run a systematic analysis in which we test statistically the association between the cell line sensitivity, so the IC50, and their characteristics. So it includes the mutated gene that come from whole exome sequencing, the amplify or deleted gene that come from SNP6 microarray, or the expressed gene coming from transcriptome analysis. So I will present the results that we obtained for nuclein 3A. So first we search for the mutated gene associated with sensitivity to nuclein 3A. We run a gene per gene analysis, or mutated gene per mutated gene analysis that tests the difference in mean IC50 in the group of cell line mutated for a given gene to the mean IC50 value obtained for the cell line wild type for the gene. So on this volcano plot, you have reported the results of this analysis with in X the difference between the groups mutated versus non-mutated, and in Y level the level of significance. The green gene are the gene for which the association is positive, so it means that it predicts sensitivity, and the red gene are the gene that predict resistance. And we see with this analysis that TP53 was by far the most significant genetic event explanatory for the resistance to nuclein 3A. Following this analysis, we run a differential gene expression analysis in which we search for genes with expression. We search the gene that have an expression level associated with sensitivity to nuclein 3A. Again, it's a gene per gene analysis. We test more than 21,000 genes, and we test the difference in expression between in expression level, in the group of cell lines sensitive to nuclein 3A versus the ones that are resistant. So on this volcano plot, you have reported the results with the of the differential gene expression analysis. In X, you have the fold change gene expression difference in between the group resistant and sensitive, and the P level of significance in Y. So with this approach, we have found a total of 162 genes significantly associated with sensitivity to nuclein 3A. In blue, here you have the genes that are positively associated, so it means the gene for which high expression correspond to high sensitivity to the compound, and in yellow, the ones for which the high expression correlate with resistance to the compound. And we can see with this analysis that the top each gene differentially expressed and associated with nuclein 3A is the target of nuclein 3A, MDM2. Next, by using these 162 genes, we performed an unsupervised hierarchical clustering. So this is an analysis with a two-side correlation. On one side, you classify the cell line and you aggregate them according to the similarity in the expression of this 162 gene. On the second side, you aggregate the 162 genes according similarity of their expression patterns across the 140 cell line. This approach allowed to delineate 3 clusters, in blue cluster 1, cluster 2 violet, or in green cluster 3, and we can see that this approach allowed to delineate clusters that have distinct response rate to significant distinct response rate to nuclein 3A. In the first cluster, we observe a response rate of, we have 64% of the cell line sensitive to nuclein 3A. In the second cluster, only 14, and in the third cluster, 22%. So similar approach for biomarker screening were tested or were run for tazelicib sensitivity. When we search for the mutated gene for tazelicib, which for the mutated gene associated with tazelicib sensitivity, we retrieve with our approach and with our data that the most significant gene was PIK3CA. So PIK3CA is the target of tazelicib, and it's a well-known gene with activating mutation that is cancer, that is a driver for many cancer types, and it's also a known, well-known target for the compound. In this screening, we have also identified for tazelicib, when we analyze gene amplification and gene deletion that could be associated with sensitivity to the compound, we identify one of the significant markers was RB2 amplification. It means that cell line amplify, so cell lines that are mainly breast cancer cell line or gastric cancer cell line associated with an RB2 amplification were those with a certain sensitivity to tazelicib. These results with our database, we tried to validate it, and we investigate a second data set that contain the second data set of tazelicib data. This is the GDS-C2 dataset from the Sanger Institute, in which the tazelicib was tested in more than 600 cell lines, and we can see that among the 20 cell lines with RB2 amplification, 12 of them were sensitive to tazelicib. So it's a way to validate the data that we generate with the ProLiFiler. So in total, with this poster together, we have shown and validate that combining the reaction biology results with the ProLiFiler to the data available on the cancer data miner help to gain insight in the potential of novel anti-cancer agents, and also to prepare the next step of development. With the ProLiFiler, we have seen that we can determine the potency of the compound and already receive information regarding the indication for the treatment. With the MOA finder, we have seen that it's possible to retrieve the mechanism of action, and for many novel anti-cancer agents, this mechanism of action is unknown. And it also this analysis also allows to identify drugs with similar anti-tumor activity, which can be helpful in case of thinking about combination study. At the end, we have seen that the molecular data we have available allow an early identification of molecular determinant of sensitivity to anti-cancer agents. And I will be very pleased to take your question. Thank you.
Thank you very much, Vincent. Excellent. So I've had some questions coming in while you've been presenting. I think, you know, really in-depth and extensive study that you've presented to us there. I think a key question from Anya would be, could you give us a feeling for the timeline required for such a project to complete that investigation?
I guess, Jan, you want to answer to that?
Yes, up front, of course, we would need the cellular data, and these usually take a month to generate. And in this ProLiFiler format, there is the possibility about every 6-7 weeks that we offer this and collect compounds from different clients. And by putting them all together, processing them together, that generates an attractive price, which then is being run, and usually takes like 4 or 5 weeks to have the IC50s. And then these go to the customer, and if they say, okay, that's interesting, that is an interesting profile, or what would you say, then that goes on to 4HF, to Vincent, and you.
Yeah. So at 4HF, what we try, so we have several discussions with the customers, but when we have defined the cutoff to determine what is sensitive, what is resistant, when we have all the IC50, we try to generate all this analysis in a standard report and to deliver it in a month, is the general idea.
Do you have to take both and the biomarker and the mechanism of action study, or is it independent of each other?
It can be independent. These are requests. We have two specific reports. One is for the mechanism of action, and one is for the biomarker. And on the top of that, if the customer would like to see other things, we can tailor a bit the study for him.
Okay, thank you, Jan and Vincent. I can see somebody typing, but in meanwhile, I have a question from Sebastian. Following on from these studies, are we able to make recommendations about the next cell lines, models to use in vivo proof to prove the identified biomarker, and then leverage that, moving forwards in vivo experiments?
Vincent, in terms of recommendations, it would be your turn on the one hand, but on the other hand, the practical stuff would then be done at the labs, of course.
Yeah. From what I know, from my experience in that, I know that if you have a certain sensitivity in vitro, and you test this cell line into a CDX, so cell line derived xenograft models, so an in vivo models, you should be able to retrieve the same anti-tumor profile. I have seen it several times. Normally, it will correlate.
Yeah, and at Reaction Biology, we have the possibility, of course, to then focus on certain cell lines that pop up in the screen, and which and to reconfirm, of course, this is a screen, and it is, of course, just been one experiment. If there is more information to that or needed, then it is wise to reproduce for selected cell lines. That would be done in a free choice format, and then we can follow this up with other in vivo studies, as you said, xenografts of human cells derived from cell lines. We also are about to fuse with another company that allows PDX models. We will introduce this information into the database from 4HF, and so we would also have PDX models in not too far away time.
Okay, that's good, gentlemen. So some more questions coming in now from the chat. So from Jeanette, where do you take the RNA-Seq mutational data from? Same for the drug sensitivity data for other drugs? Is this publicly available or from data sets?
Yeah. So for the time being, what we have mainly in the database validate, we have whole exome sequencing data for mutation. We have SNP6 data for chromosomal aberration. And most of the gene expression profile we have derived from microarray analysis. And this data for a large part of them were analyzed by 4HF. And for data that are missing, we also use publicly available database. Regarding the drugs, it's the same story. It means that in our development, 4HF has tested more than 200 compounds for anti-tumor activity. It was tested in vitro. And so it was the basis of our database. And on the top of that, we added several other databases publicly available.
Okay, thank you, Vincent. And then I appreciate we're right on the hour, but just a very quick question from Anna. Would the pipeline be also applicable for first-in-class inhibitors where comparable inhibitors and their databases are not available?
Again, please.
So would the pipeline be also applicable for first-in-class inhibitors where comparable inhibitors and their databases are not available? So very novel molecules.
I guess it is, if you have a very novel molecule, then the biomarker analysis would be the first thing to look at, right, Vincent? While it could be that there are similarities, many compounds are not necessarily completely clean inhibitors, and such a study could show up similarities to other compounds that are in clinics already or are in research already. Yeah, but what do you say, Vincent?
Yeah, it's the point is that when you have a novel molecules with really a well-defined or a clean molecules that really target really a specific gene, and when it's really clean, you are sure that your biomarker analysis will tell you what is the mechanism. Now, when you have molecules that target, I don't know, several kinases, when you start to run your biomarker analysis, it's much more complex. And for the mechanism of action, it's the same. But I think with the drug data we have in the database, when you test a novel molecules, it's always interesting to check what are the other drugs that correlate with that, which profile correlate with that, to try to better understand what are the player of the anti-tumor profile.
It's also, I think, if you have a compound where you do not know the mechanism of action, right? So if you have from natural sources just extracts, and you've purified some structure from there, and it does something phenotypically, but you don't know what it does, and you need to do deconvolution then of the target, then this strategy using the comparison to other compounds, for example, or the biomarker analysis, valuable first step for deconvolution. Of course, it would, or target deconvolution. Of course, it would require confirmation and validation on experimental basis again, but that would be also an excellent application for that, I think.
Okay, thank you very much, gentlemen. We've got one last question, but I'm conscious that we're now coming up to 5 minutes past the hour. And so I'd suggest that we capture that, and I'll email it to Vincent and Jan and ask you to reply. Is that okay? Or do you want to have?
We can have this question.
Okay.
Still.
Yeah.
It looks like it might stimulate a bit of discussion, but here we go. So
No problem.
From Claudia, what are your current thoughts on why the KRAS inhibitor BI3406 did not show the expected profiles in the cell panel?
That's interesting.
Yeah.
Yeah, it's we were actually testing in that field too, and we think that major reason is that there is strong evidence that these compounds are not as active in the 2D setting as compared to the 3D setting, leading to different sensitivity, different gene expression profile. So currently, we're testing these compounds also in soft agar panel. So we have a panel of 100 cell lines growing a soft agar that leads to 3D growth on the one hand, and to longer incubation times on the other hand. Usually that's like 7 to 14 days of incubation time. And we already see significant differences, and that could be applicable, especially for the biomarker study, because we have for all of these cell lines, or most of these cell lines, we have the gene expression profiles. But on the other hand, it will not be possible to directly compare that to the mechanism of action comparison, because these data that we use in the database have all been generated in 2D. And so there is a that is what we think is the reason, and we've been in touch with the researchers that worked originally with BI3406, and they also state that it is a critical part to check that in 3D.
Okay, good. That seems like a good place to finish up. Thank you very much for all your questions, and to Jan and Vincent for presenting the poster.
You're welcome.
Thank you very much.
Okay, I'm going to stop the recording now, and thank you for attending.